Hello, my name is Maya Shoemaker. It is July 8th, and today we will be discussing the prokaryotic-eukaryotic interaction in the urinary system. Let's go check it out. We'll be going over the prokaryotic-eukaryotic cell interaction in the urinary system, specifically looking at the uropathogenic Escherichia coli, also known as UPEC, and the interaction with the bladder epithelial cell. So looking at this prokaryotic cell, there's a couple takeaways that are important for the interaction, specifically the size being so small, 2 micrometers, and the type 1 fimbriae shown here with its blue FimH adhesion on the tips. The fimbriae is hair-like projections, and the FimH is a protein that specifically binds to mannose-containing receptors on the surface of the epithelial cells. And this will be important for the attachment interaction between the prokaryotic and eukaryotic cells. Now, in the eukaryotic cell, it has all of the same organelles as a normal eukaryotic cell. However, there are a couple differences as highlighted. First being the distinct feature of the multinucleated cell. So it has 2. This is for umbrella cells because it enhances their ability to maintain and repair the protective barrier of the bladder, especially under the mechanical stress of the bladder filling and emptying. So, and the other distinct feature is this apical membrane. These are these hexagonal plaques, which create a rigid protective barrier that prevent toxins and pathogens from penetrating the bladder wall. It is formed by these uroplankton proteins, which are essential components of the apical membrane. And these are where the FimH little guys will have the receptors which are found on the uroplankton protein. So it all ties in together. Now, like I said, the plaques all around the cell are important for the bladder integrity, but they are a high affinity binding site for UPEC, and they give the bacteria a foothold in the initial infection and the attachment stage. One thing to note is the difference in size. Like I said, the prokaryotic cell is only 2 micrometers, whereas because of the fluctuation of a bladder, the umbrella cell can fluctuate between 50 and 100 micrometers. And this is important for later in discussing their interaction between the two. Let's move on to the next
Diving
step.
deeper into the interaction between UPEC and bladder epithelial cells, specifically the superficial umbrella cells shown here, you can see the attachment that UPEC does by binding to the apical membrane through the type 1 fimbriae. It begins an invasion and intracellular replication, which proceeds to go to intracellular bacterial community formation, IBCs. These IBCs are clusters of bacteria that replicate within the host cell, forming bio-like film structures. As the IBCs grow larger, they can disrupt the stability of the host cell and eventually lead to the shedding from the bladder lining. When this occurs, UPEC is released into the surrounding environment where it can continue to spread the infection. Within these communities, IPEC can also enter a dormant state, allowing to evade the host immune response and survive antibiotic treatment. This is due to the smaller size and adaptability of the UPEC size and the ability to hide in the much larger eukaryotic cells, increasing their persistence. That is it for the interaction between UPEC and bladder epithelial cells and the prokaryotic-eukaryotic interaction. Thank you for watching.